Hello, everyone. I'll wait just a minute before we get started. Hi, Mikey. Hi, Abigail, Danny, Roland, Michaela, Tori. I guess that's everybody. All right. And yes, I have not left my office yet today, which is not fun. But I'll get to go home when we're done with this. So, any pressing questions before I sort of go through some? I just had sort of an itinerary of stuff. So I was going to look at the study guide. Let me actually post this in Teams right now for you guys. I'm going to post that document. Let's see. So this is the document for review session. So any questions before I sort of get started? All right, so I just posted something in our Teams channel. I'm going to be sharing my screen and looking at that document anyway, so you don't need to necessarily have it open. So let me get over here and I'm going to share my screen. All right, so here's a document that I shared. Basically, the exam is 4 total questions, but you know that that's sort of irrelevant because questions have multiple parts. But they're generally broken into these 4 groups. So they're, and again, 100 points. So it's kind of a little over a quarter is for this first one, which is all about the details of glycolysis. So you will be asked several questions. Every time there's a blank, it's essentially 1 point. So you can sort of get a gauge of how many different things I'm asking. I gave you the first one just so you sort of have an understanding. It's like ATP production occurs at these steps. And you'll see in here, it says in parentheses, enzyme. So there's 2 places that ATP is produced. So you need to put the enzyme names in there. So again, I'll either be asking you a step number, an enzyme name, or a substrate product or cofactor name. So it'll say S name, P name, or CF name. Okay. So you're going to want to know, glycolysis pretty much in and out. I know some of you have used that website to practice, whether you download and sort of have the structures and all of those pieces together. However you want to study, there's only 2 pathways that we're doing this with. It's with glycolysis and then the citric acid cycle. And largely that's because a lot of disease states that we're going to talk about feed in or out of these 2 metabolic pathways. So we want to know them pretty well. So that is that question. So what I'm going to do is I'm just going to go through these 4 pieces, see if you guys have any questions on that, and then I'll do what I've done in the past, which is sort of just go over the review sheet, and there's really only one learning unit here, and then talk about if they want to go over any homework questions. And I will tell you everything that you need to know here is directly from or directly related to stuff that's on homework, notes, not really case study as much. The case study is just sort of an application of stuff. So that's the first question. Remembering, again, you're going to get 10 minutes of access to your notes. So don't, worry or spend a lot of time if you got stuck on something, especially if you know where to find it in your notes. You'll be able to go back. You certainly won't be able to do the entirety of that section in 10 minutes. That 10 minutes does go by pretty fast, or at least that's what students in the past have said. Then there's about a third of the exam is relating to biochemistry that's beyond glycolysis, but still related to glucose and glucose metabolism. So thinking of things that you could either watch from a video standpoint, find as a section in your notes or your homework, these are sort of some key pieces to sort of study these. Um, the story of glucose, right? There was a lot that we talked about with structure of glucose, numbers that are important to glucose, things like that. Also be able to think about what we did with pyruvate after we were done with glycolysis. We spent some time talking about the pentose phosphate pathway. Um, think about, and these will be things that we'll build on, think about places that we have talked about generating important metabolites. And so think about where we've generated carbon dioxide in processes that we've learned thus far. We'll continue to sort of expand and explore that as we learn more pathways. But knowing that that's one of the main waste products that we produce in our fuel equation, it's important to sort of think about where we produce that. And then we did a good bit understanding the structural regulation of glycolysis. So some details related to that. And as these pieces come back up in when I go through the study guide or we look at our notes, I'll kind of remind you of these pieces. So that is the bulk of the exam of stuff that 100% it will be in your notes. It'll be in homework, something like that. Then there are two questions that are going to ask you to apply knowledge to a new situation. So there is a 20-point multi-part question that is going to be related to concepts that you learned about when we were studying PFK regulation and 2,3-BPG. And you're going to want to apply them to a fictitious metabolic pathway. Okay. So make sure that you understand how PFK regulation sort of is affected by substrate concentration. We had those plots. And then we had the story of 2,3-BPG and how binding that metabolite changed R state and T state in a different system, but how sort of these pieces work together. So if you understand how these pieces work with this, you're going to be asked to apply it to a fictitious metabolic pathway. Okay, highlighting it's not about glycolysis, it's not about 2,3-BPG, it's not about oxygen binding to hemoglobin. But if you understand that, you should be able to translate that knowledge onto this fictitious metabolic pathway. So that's a question that's worth about a fifth of your exam. And then the last question, you will be able to choose from two different things. There are, I think you're sort of maybe a 50-50 split of people that would be fine if I left mechanism in and people that are grateful that I took it out. So this was a question that was there last year, so I'm going to keep it. If you hate mechanism, then this is the question. The B part is what you're going to do. There's some questions related to thinking about how you interconvert molecules that are part of glycolysis. So being able to think about structures of glycolysis and maybe drawing mechanisms that would allow us to think about that. So again, if you are somebody who's sort of not mechanism averse, that might be something that you could sort of figure your way through. If not, then think about studying how to detail an enzyme active site, because that is going to be an important skill as we sort of go forward here. I'm going to give you an enzyme that you have not seen. It is related to glucose metabolism, though. So you have learned about this chemistry. You just haven't seen the enzyme active site. And you're going to have to look in the enzyme active site and like, what do you think this amino acid is doing? What molecule is here? What chemistry is going on? Detail a step that I ask you to take a look at. Okay. So especially since I sort of give you, blanket access to your notes, I really want to see that you can learn something and apply it to sort of a new system. So that's what these last two questions are about. So I'm going to pause there for a second, see if anybody has any questions on these before I sort of go and start doing a little bit of a review of content and sort of highlight how it sprinkles in here.
Just for clarification, that first one, you can hear me, right?
Yep.
Okay, cool.
Yep.
That first one for ATP production occurs at these steps. You want the step
Mhm.
number and the enzyme in that step?
So this one, I'm asking you to provide the enzymes. You'll always see in parentheses what I'm asking you for. So just as a hint, as you're doing this problem, some of them will say enzyme, some of them will give you a number in parentheses, some of them will say product, or some of them will say substrate. So what I would encourage you to do is you go through these problems. Like if you can't remember what step number it is, that's fine. Write down the enzyme name or the substrate or something that you remember about it. And then when you have access to your notes, you can sort of go back and fill that in. Okay. But do make sure that you read and see what I'm asking for in that problem. Okay. Anything else on this? All right. I'm going to go over to Blackboard here. And let's see. I think I had pulled up here. I'm just going to show you real quick. These were the slides from that first learning unit. You're never going to be tested explicitly on something related to this, but I do want to highlight things that are sort of we're seeing again in this learning unit. Not anything specific on fundamentals, but of course I would assume that you still remember how to determine protonation states and pH and temperature. Obviously, this piece was an important part, thinking about covalent modification, kinases, phosphatases, need state, fed state, and so forth. Not too much with thermodynamics. Obviously, we're talking about carbohydrates, so make sure you know these details about carbohydrates. No protein structure or lipid structure at this point. Carbohydrate structure. I would make sure you know the structures of important carbohydrates as they relate to things that we've learned about in glycolysis. Nothing with nucleic acids. Again, reaction mechanism, there is a whole section of notes as well as videos on reaction mechanisms. But if that is not your cup of tea, you can just choose do the other problem. Not really reaction coordinates in that this learning unit. We did, obviously, when we talked about 2,3-BPG, think about the Perutz model and equation. So that's a piece that sort of found a home here. And then, this expanded fuel equation should always be in the back of your head. This is essentially the entirety of Biochem 2, but we are learning about all the different processes that will metabolize different fuels. So right now, the fuel is glucose, and we've studied glycolysis. So think about how different pieces here feed in. We synthesize some NADH. We do synthesize some CO2 if we talk about using G6P in the pentose phosphate pathway. Nothing on Michaelis-Menten kinetics here. So not too much to say there, but always remember that there is content that is in that first unit, which is just meant to remind you of things that we'll see again. So let me go here and I'm going to pull up the study guide. If I can find, was there a study guide that you guys saw? Maybe it's over here. Hang on. There it is. Study guide. And don't worry that it says 2022. It's still the same content. All right. So I'll sort of run through this quickly, then we'll spend some time looking at notes and then homework. And then if there's anything either before or after that, don't hesitate to sort of interject here if there's something you want me to kind of highlight. So I gave you guys some details about those 10 steps of glycolysis. For any of these, I'm going to ask you step number, enzyme name, substrate product, cofactor name, chemical structure. Unless specified, you can either choose to provide the full name or the abbreviation. That does not matter to me. Okay, but that's sort of a fairly detailed section. Fundamentals information, you're not going to like do the entirety of the fundamentals piece, but those little pieces are interwoven sort of as we went through here. That's kind of related to the first part, knowledge of both full name and abbreviations for metabolites or enzymes, needs for cofactor and co-substrates, right? Hopefully when you look at these, you're like, oh, where did I always see magnesium? I saw magnesium anytime I had ATP. And why it was there is magnesium helps to balance that super negative charge we have in that polyanion that is ATP. That helps ATP do its chemistry, because oftentimes we're going to need the phosphorus of ATP to be electrophilic, which means it needs to be electron poor. That's not going to work when you have an abundance of negative charge. So magnesium helps to balance that. I'll show you on a slide when we go look at our notes where that is. Again, being able to think about where we generate certain products. Obviously, the rate controlling step for glycolysis is the only pathway that we're talking about right here is PFK. Know where we have large negative delta G values, hexokinase, phosphofructokinase, and pyruvate kinase, first, third, and 10th steps. Everything else is near equilibrium, which means our delta G values are close to 0. And then be able to think about the types of chemistries that are happening, right? As you are learning glycolysis here, hopefully you're thinking about, okay, hexokinase, that's a phosphorylation step. Kinases are using ATP to transfer a phosphate onto an OH group, right? So be able to think about the different kinds of chemistries, right? When we have functional group migration, I know that's going to be a mutase enzyme. So there's lots of things that we sort of learned about with just if you learn some of these pieces, they're going to make learning things later easier because you're not individually trying to memorize all of these different things. Like if you see a dehydrogenase enzyme, hey, that should tell me I've got redox going on with that, which means I'm probably going to need some of these B vitamins as well. So just some of those big picture pieces in terms of types of reactions, and then as they apply to things we've learned in glycolysis. Again, details of enzyme active sites, right? Especially if there's a couple that we talked about unique roles for amino acids. So I would do your best to study your own notes. You can have access to my notes, right? Because many of you, if you're taking paper notes, sometimes it doesn't work as well if you need to have a picture on there. But be able to think about some details of glycolysis. Questions on that?
I have some questions.
Yeah,
Um,
sure.
when we're talking about inorganic phosphate, is that just adding a phosphate group to, like, where's that come in? I know it's around the same places where the B vitamins come in, but what role does that play?
So the only time we see inorganic phosphate in glycolysis is we see it in step 6. It's one of the co-substrates that we need in order to phosphorylate GAP to make 1,3-BPG. Did that whole bunch of alphabet make sense? So it's sort of like we don't always see phosphate as inorganic phosphate being added to something. That's sort of a little bit of a rarity. Oftentimes it's a phosphate that's coming from ATP or coming from something else that's already high energy, so can easily transfer its phosphate. So that's a little bit unusual, but we also see that we're doing, that's a kind of an involved step. It's got a positive delta G value. There's a lot going on in that conversion of GAP to 1,3-BPG. So does that help with that question? Did you have another one you said, or?
Yeah, what when it comes to ender versus extragonic, what
Mhm.
part does that correlate to?
So endergonic is a positive delta G. So it's just another way of saying a positive delta G. So be able to think about, there's only one, reaction, we were just talking about it, that has a positive delta G value, reaction 6. It's coupled to the very exergonic reaction 7. And then everything else is sort of near equilibrium except for the hexokinase reaction, reaction 1, and then pyruvate kinase reaction 10. You do not need to know numbers at all. The only number that's sort of relevant is that we've got 30.5 kilojoules per mole for anything that involves ATP hydrolysis. Any other questions on this? All right, so some details on the beyond glycolysis piece. Right, know the structure. I would know the structure of glucose. You should know the structure of glucose, right? Know the levels of glucose. We learned 3 ways that we can think about glucose levels, right? You can do a snapshot with a blood glucose level. So remember either 90 milligrams per deciliter or 5 millimolar, right? Those are 2 ways. And you had a math problem on your homework that allowed you to show that those were equivalent concentrations. And then hemoglobin A1C, right? Around hemoglobin A1C of around 5 is going to be sort of a normal average blood glucose. So I do want you to remember those numbers and how we measure those. So, right, blood sugar is just, literally a snapshot of the concentration of glucose in your blood. Hemoglobin A1C, that is a percentage of glucose that's percentage of hemoglobin that's decorated with glucose. So the more sugar that you have in your bloodstream over a 3-month period, the more decorated your hemoglobin is going to be with glucose. So hemoglobin A1C values are a percentage of hemoglobin that is decorated with glucose. Okay. And then big picture, we talked about how we remedy either high or low blood sugar. So we're starting to get into the weeds right now with our glycogen metabolism chapter. So it's easy when we have like a little bit of a gap here before we have an exam to sort of get the waters a little bit muddied with, like, we're talking about things with phosphorylation cascades and stuff like that. Not what you need to know in terms of relevance here. You just need to know that we break down glycogen or synthesize glycogen, and then we can do gluconeogenesis. We haven't learned the details of those yet, but we did learn about sort of those top-down and bottom-up approaches. So sort of a great video for helping with that. Oops, need to get into this folder here, right? That's what the story of glucose is all about, is sort of those different pieces. Fates for pyruvate, right? There's 3 possible things we could do with pyruvate. It can go into pyruvate dehydrogenase chemistry in the mitochondria, right? We can have homolactic fermentation. And even though it's not something we can do, there is alcoholic fermentation. Sort of, we learned about that. So we can sort of look at those pieces. In parentheses here, if you didn't sort of know this with these study guides, these correspond to the homework questions that are relevant. Now, there's only 1 unit here, so it's like just 2.4 is the 4th question on Unit 2's homework. When we start having maybe 2 learning units that we have on an exam, that will indicate which learning unit it's from. So again, I will show you and highlight where these pieces are in your notes as we go through them. Also be thinking about for lactate, right? What we do with, so just to finish up the pyruvate piece, what we do with pyruvate is largely dependent on whether there's oxygen around, aerobic versus anaerobic sort of metabolism. The same is kind of true with lactate, whether we are able to deal with lactate immediately if we have oxygen around, or have to sort of delay and deal with it later. So we'll sort of look at that on that slide. When we do talk about pyruvate decarboxylation, again, that is something that happens with alcoholic fermentation. We can't do, but you do need to know the biochemistry for it, because that's what happens when we brew beer or any alcoholic beverage. It's problematic, right? Because alpha keto acids do not easily decarboxylate. Beta keto acids do. We'll look at that slide in a minute, right? We've got TPP, we've got vitamin B1 that's going to help with that. You guys will see, and hopefully you had a first exposure in Gen Chem 1 when you had to learn your vitamins, and you've probably memory dumped that from your brain, but hopefully it's not completely wiped clean, because you're going to see all of those B vitamins again. And they'll probably stick a little bit more because we'll have a lot more context for them. So vitamin B1 and as TPP, we need for any decarboxylation of an alpha keto acid. Then we talked about the pentose phosphate pathway. So an offshoot of glycolysis, we can either siphon off G6P or F6P and GAP. So lots of places we can either siphon off and go down, or sort of siphon and come up. Know the 3 stages of the pentose phosphate pathway. We sort of have the main 2 products that we make. We make NADPH and we make R5P. Right? Those are really important, largely if we're doing cell division, things like that, where we need to have lipids for that purpose, and we need to be replicating our DNA. So that's why the pentose phosphate pathway is a pathway that's often commandeered by cancer cells and things like that, because it's going to allow them to make sure that they can make all those building blocks that they need. Rate determining step of the pentose phosphate pathway is G6PD, so glucose 6-phosphate dehydrogenase. And again, the entry and exit points from glycolysis are G6P is how we're going to exit to go through the G6PD chemistry, but then we can also siphon up from the bottom. If all that we need to do is make ribose 5-phosphate, we can siphon up through stages 3 and 2, kind of working backwards by siphoning off F6P and then GAP. So we'll see these again when we go through and just kind of run through our slides real quick. So especially as we start learning multiple pathways, processes, enzymes, as we start seeing branch point metabolites, try to see where those feed into existing pathways that we've learned about. So again, we really haven't learned too much right now, but we've talked about NADPH. So not only is it important for lipid biosynthesis, but we talked about its importance in glutathione regeneration. This is really the only point that we've talked about carbon dioxide production in us is through G6PD chemistry. We can generate carbon dioxide if you talk about alcoholic fermentation, but again, that's not a metabolic pathway that we see in humans. And then again, lactate. Lactate's one of those dead-end metabolites. And so up here, we sort of talked about what are the fates for lactate that we can do. And then the one cascade that we sort of talked about, just, and I have cascade in quotes because it's not really a cascade, it's just a thought process in terms of why does a person who's G6P deficient more subject to hemolytic anemia? So we're going to see a lot of these where you're going to need to connect an upstream piece with a downstream, often physiological process. So I know a lot of you are taking physiology, so you might find some overlap with things that we see here, but often we're going to have things that we may go into a little bit more detail on. So I'm going to pause there for a second, see if there's any questions on those pieces. And again, when we go back through and look at our notes, you may find that you have additional questions that we can address. All right, last little bit here, and I'm not going to say too much on these because I'll address them as we get to those relevant slides. Obviously, reaction mechanisms. You know, if you feel like you're comfortable with it, take a look at the reaction mechanisms that you see for glycolysis and see if that's something that you feel like you could apply in a situation. Talking about how you regulate metabolic pathways and identify flux control points. Really, you're just looking for large negative delta G values. Those are going to be good targets for regulating metabolic pathways. Related to that are substrate cycles. You often have substrate cycles where, like for glycolysis, steps 1, 3, and 10, that's where we have large negative delta G values. So if we want to reverse that sort of chemistry, we can't use the same enzyme and we can't use the same pathway because it was downhill. So we have to choose a different pathway and thus a different enzyme, and doesn't, specify that one direction has to be energy input, which one, but you are going to have one of those pathways will have to input energy. We can't go downhill both ways. So substrate cycles allow us to interconvert metabolites where they are generally flux control points. So we're going to have a rate controlling enzyme on the catabolic side, and then an opposing rate controlling enzyme on the anabolic side. I'll revisit these when we get to those slides, but when we say interpreting structural data, interpreting enzyme kinetics, and interpreting substrate binding curves, that has to do with understanding PFK and then understanding 2,3-BPG. And you will need to know, little bits of that for things that you'll see on the exam, but you're not going to see one of those entire questions, but you will need to apply your understanding of that to understand a new system. So those are sort of some of the big questions and concepts. And then sort of we talked about glutathione regeneration and how that connects with malaria, right? Because somebody who is more subject to hemolytic anemia is less likely to be infected with malaria because that malarial parasite can't sort of grab a foothold. We talked about the connection of 2,3-BPG with hemoglobin function. And then don't worry about this last one. It was just, we had a slide where we sort of talked about, hey, if you wanted to study whether something was happening stereo specifically, right? You can use deuterated compounds, right? Deuterium is a modified hydrogen. It's heavy hydrogen. And so that is going to be spectroscopically different. And so you can sort of follow pathways and understand sort of what's happening in a metabolic pathway by using isotopes. So you're not going to see a question on that, so I wouldn't worry about that, but that's something that, as it's relevant, we'll sort of see throughout the course here. So I'm going to pause there for a second, and then I'll head over in just a second to look at our slides. Any questions? All right, so I'm going to go to our slides, and if there's something that you'd like me to sort of pull out for like more discussion, let me know. Again, there is a story of glucose slide, but there's lots of information that's on here in terms of things that you sort of want to know. You know, anything hemoglobin A1C that's in the 4 to 5 range is going to be sort of a good level. Again, 90 milligrams per deciliter, 5 millimolar, be able to draw the structure of glucose. Sort of know these top-down and bottom-up approaches to remedy low blood sugar levels. Know your glucose transporters. We've seen those before, right? GLUT2 transporters are in the liver. They're always present. GLUT4 are insulin sensitive. So again, really important slide. If you feel like you need that refresher, that's why I have a lot of these complicated slides. There's videos that you could rewatch. Anything on that one? Not going to go through fundamentals. I think everybody pretty much aced the fundamentals when you did it. There are some key things that you might see as they relate to those glycolysis questions, right? Here's an important metabolite that you need to regenerate. So you might see a question about what's the enzyme responsible for generating that important metabolite, or what step does that occur at, or and so forth. That was fundamentals stuff. Right, there's a few videos I'm just going to kind of highlight here. There's a glycolysis walkthrough. This was from when I was teaching during COVID, and so we actually didn't even come back to class that spring semester for like the first week and a half because it was the height of Omicron, and so we actually started this semester late. We had everybody start remotely, so I was doing glycolysis from my office at home, and this ended up being a really useful video to do a walkthrough because I wasn't able to sort of set my table up and sort of show structures and so forth. So you might find that useful as a way to sort of review glycolysis. I know some people have highlighted that they love this game to go and play. So if you haven't done that yet, you might find that useful as a way to sort of quiz yourself. If anybody finds anything else, do let me know so that we can sort of share it with your classmates. If there's something that you find that's helpful to help have you learn sort of the story of glycolysis. So as you go through these slides, right, if you're on the hexokinase slide, quiz each other, ask other people, like what's going on with step 1? Okay, step 1 is glucose converted to glucose 6-phosphate. All right, what are the cofactors that I need? Cofactors, again, generally are metal ions. I'm going to need magnesium for that. You can see here why we need magnesium if we're doing anything with ATP. It's going to sort of help make that phosphorus more electrophilic so that we can, transfer that phosphate group that we're going to have on there. So I'm not going to go through the entirety of glycolysis here, but as you look at each one of these slides, make sure that you can sort of understand and see all of those pieces: substrate name, product name, enzyme name, kind of chemistry that's happening. And that's why there was a homework question, right? Because I didn't put the homework key up here. And I think there's actually, this is specifically for that homework problem. Like this is the money piece. Like it tells you all of those things, right? What's happening in that step, the substrate, the product, the enzyme name, if there's a coenzyme or co-substrate that's necessary, the chemistry that's going on. Okay. So
why
not gonna
does step
yeah.
sorry. Why does
Yep.
step 10 need both magnesium and potassium, where the rest of them just need magnesium?
Let's see. So where, and I might need to know where you see that. Oh, it's on that. Okay, let me open that up. I know exactly what you're looking at. I don't know that. And because you found a little Easter egg that I was not even aware was there, you don't need to know that.
Okay,
My
it was on the homework key too, so I wasn't sure if that was
No, and
something
that's
we need to
probably
know.
because I just pulled it from here. And I'll have to think of, oftentimes, potassium is just there as a way to help stabilize enzyme structure. But I'm gonna have to go do a little searching because either I haven't talked about it in long enough where I've forgotten, or I'm not certain. You do not need to know about potassium there. Good eye observation here. All right, I gotta move my stuff down. All right, any other questions or details on different enzymes or chemistries that we have here?
Are we going to have to be able to draw any of the structures?
You might. Yep, I'm not going to ask you to draw all of them. There's not time for that. But one of those questions that's on that first part here, okay, you may have to provide a chemical structure. It's not for all of them, but I am going to ask you a chemical structure for something. Okay. So if you were given a name of something, hopefully you could sort of work your way through what the structure is. Okay, you have to know glucose. We've had to know glucose forever. Fructose, we've sort of drawn enough. And then everything else, you might be able to use some organic chemistry skills to sort of try to deduce what that structure is if you don't remember. All right. Then we sort of got into what we do with pyruvate, right? And there's 3 arrows here because we have 3 options, right? This is an important piece that was on your homework, kind of saying here's the different things we do with pyruvate. We can go into 2 different fermentation processes, right? This is homolactic fermentation, right? So we've got LDH, lactate dehydrogenase, and then we've got alcohol dehydrogenase. So be able to sort of think about, it's not written in here, but be able to think about the different enzymes that are going to utilize pyruvate and then the products that you might make, right, for those steps. So being able to put those pieces together. So again, we've got lactate dehydrogenase. That's something that we do. Obviously, we lack pyruvate decarboxylase, but then we've got alcohol dehydrogenase doing that dehydrogenase chemistry, that redox chemistry. I want to sort of just mention here again, the what you do with lactate depends on whether or not you have oxygen immediately available or not. So if you have oxygen sort of immediately available, you can reconvert lactate into pyruvate and then continue down this aerobic pathway. But if you continue to be in an anaerobic state, you're doing some anaerobic exercise. That's why you can accumulate lactate when you're exercising anaerobically, and you will end up exporting it from your muscles. It'll increase in concentrations in your blood, and you end up sending it to your liver and says, you know what, liver, you take care of this. Okay. And so one of the things that we'll revisit later is talking about, how you can monitor blood lactate levels when you're exercising. You can use just a little finger prick test to sort of monitor what's happening. And that's a good way to sort of assess what's going on in your body and how sort of aerobically fit you are, is to sort of do lactate testing while you're exercising. This again is an important piece of highlighting you cannot decarboxylate alpha keto acids because anytime you're doing decarboxylation chemistry, you're going to be taking a carboxylic acid group off. Chemistry is always the same, right? We're going to form a pi bond here with that lone pair of electrons that's on the alkoxy oxygen. Can't have 5 bonds to carbon, so the bond wants to break. This cannot happen when you have an alpha keto acid. Look what's happening over here over the beta keto acid. We can do that because we can, even if we start out localizing the lone pair of electrons on this carbon here, it's adjacent to a carbonyl so that we can resonance stabilize through there, right? So that's a really important thing. We are going to see this throughout the course as we talk about generation of CO2. Oftentimes, generation of CO2 is from decarboxylation of a metabolite. And so we will see a lot of times when we decarboxylate alpha keto acids, but We also know that we're going to need to see vitamin B1 with that. So just like if you see something with a kinase, you're going to need magnesium for that enzyme to work. If you are decarboxylating an alpha keto acid, you're going to need TPP, which is used to make made from vitamin B1. All right, and then this was just that slide reminding us like, hey, if you want to look at a little bit in terms of how enzymes work, you can substitute a hydrogen for a deuterium, and because they're spectroscopically distinguishable, you can actually do some really neat spectroscopy, whether it's NMR or things like that, to sort of follow what's going on in enzyme active sites. All right, I'm going to pause there, see if anybody has any questions on sort of glycolysis and sort of those post, after glycolysis pieces. All right, so importance on this slide was identifying flux control points, large negative delta G values, right? This is going to be sort of an important concept to think about, okay? We ruled out, ideally, if you're going to control flux through a pathway, have your control point right at the beginning, right? The only reason we eliminated hexokinase as a putative control point is we know so much feeds in and out of glycolysis at G6P. So it didn't make sense to have our control point upstream of that. So it's not to say that you won't have metabolic pathways where the first step is the rate controlling step, right? Pentose phosphate pathway, the first step is the rate controlling step. But you have to think about the entirety of what's going on in the pathway. And if you have a significant branch that happens downstream of where you're trying to control, that's not a good thing. So that's sort of one of those big picture things to sort of remember. And of course, controlling things at the end doesn't make any sense, okay? So that's one of those important things to be thinking about. You might be asked in a fictitious metabolic pathway to sort of help make those determinations. I don't think there's anything else to highlight from that slide. Some big picture things to think about is we start learning about some molecules that are going to be consistently fed state or need state signals. And we're going to get more to F26P in our current learning unit, but again, not relevant for the exam here. But when we regulate PFK, we're going to see its regulation from structural allosterism. So binding these molecules is going to change between R state and T state. Okay, this is not an enzyme that's subject to covalent modification. So it's not one of those that we regulate by adding or removing phosphates. Okay. So again, an important slide. There's a video on this, thinking about how PFK is regulated, right? It's regulated by structural allosterism. So binding different molecules is going to change from R state to T state. Again, as we shift a binding curve to the right, that means we don't bind our substrate as tightly. Shifting to the left means we bind more tightly. So I'm not going to go through this in detail, right? There's a video that's on there if you need to sort of go through if this was confusing when we did it in class. Kind of go back through and sort of watch and understand. This was for an enzyme activity, how we can shift curves based on whether we are adding inhibitors or adding or rescuing activity with activators. Okay, so that's sort of an important slide to understand some details from. This is an important slide to sort of understand structurally how PFK is regulated, right? We've got small molecules that bind here. They're going to change between T state and R state. You do not need to remember whether it's winding or unwinding, but when we toggle between T state and R state, that changes what amino acid residue is in the enzyme active site, and ultimately that's what's going to determine whether we increase or decrease the affinity for our substrate. Okay, so again, there's a story on these two pieces sort of together in terms of how we think about regulating PFK activity. So I'm going to pause there for a second before we get into the pentose phosphate pathway slides. All right, over the years, I've actually taken out a lot of stuff on pentose phosphate pathway. So even if you look at my homework key, you'll see that there's way more questions that are there. You should be able to find the pieces as they relate to your homework question answers, but lot fewer details that we need to know about the pentose phosphate pathway. This was introducing us to that concept of substrate cycling. If PFK is a large negative delta G reaction, that is the rate controlling step of glycolysis, going in reverse must utilize a different path and a different enzyme. And this again, in the forward direction, is going to be the energy input phase. You can see that's where we use ATP. So it's actually downhill energetically to have FPBase do its chemistry. So we talked a little bit about substrate cycling, sort of introducing ourselves to the fact that this is going to be the rate controlling step for gluconeogenesis, which we're learning about on Friday. Just talked a little bit about how substrate cycling works. At the end of the day, you're just controlling the rate or the flux. Ellie, you'd ask this question, like, what is flux? Flux just has to do with how fast flow is through a pathway. So it's really kind of a rate of enzyme activity for a forward and a reversed reaction that ultimately determine which way you're going. And I really like to use sort of like water in a hose analogy to think about this, because if you had water sort of going in the same rate in a forward and a reverse, you just sort of spin in this circle. But again, if you have a greater flow of water in the forward direction than the reverse direction, the net flow of water would be to go down. Reminded ourselves of uncoupling proteins, right? We've seen those quite a bit. So when there's something important or I want to kind of carry as a thread, we'll often have a slide that we just sort of revisit it. But this is again highlighting these 4 important steps that we know for cellular metabolism: glycolysis, pyruvate dehydrogenase chemistry, citric acid cycle, and then electron transport and oxidative phosphorylation. With the exception of taking a little divergence to do a little bit on glycogen metabolism, that is going to be pretty much half of the Biochem 2 course, is sort of going through these different metabolic pathways. Reminding you cannot interconvert NADH and NADPH by a kinase. Okay, so that's not something that happens. They're metabolically distinguishable, but they serve similar functions, right? NADPH is in its reduced form going to be used to provide the electronic glue to glue atoms together to make molecules. Specifically, they're going to be used for fatty acid synthesis, right? We also talked about its importance for glutathione regeneration. So again, here's the different pieces. I want you to know that there's 3 stages to the pentose phosphate pathway. Main products that we're going to make are NADPH and ribose 5-phosphate. Siphoning off from G6P is one way. You can sort of see that with this arrow here. And largely, anytime we snip off CO2, we're not going to be able to reverse that reaction. If all you want to make is ribose 5-phosphate, you can siphon up from F6P and GAP and go back through stage 3 and stage 2, and then sort of make that ribose 5-phosphate. So not a lot of details we learned about with this pathway, with the exception of, I do want you to know this enzyme name, G6PD. That's the rate controlling enzyme of the pentose phosphate pathway. And we sort of talked about G6PD deficiencies as they relate to an inability to regenerate glutathione. So glutathione is your biological antioxidant. So if you cannot make sufficient NADPH, that means you will have insufficient levels of glutathione, which means you are more susceptible to damage from oxidative species, right? And so we saw that sort of in this story where individuals who are G6PD deficient are going to have low levels of NADPH. They're going to impair their ability to regenerate their glutathione. They're going to be more susceptible to oxidative stress, which in a downstream way makes them more susceptible to red blood cell lysis. And that's called hemolytic anemia, right? But then we can tie that together. We know that people who have increased cell lysis are resistant to malaria. And that's why people who have sickle cell anemia, if you are a carrier and you may have increased cell lysis, but not necessarily sickling, that's why we have that selective advantage for the persistence of that sickle gene in regions that are endemic to malaria. And so the same here that we're going to see an increased prevalence in G6PD deficiencies in areas where we see lots of malaria. So that kind of wrapped up that story. Okay. We spent a little bit of time sort of talking about and reminding ourselves what other sugars we're going to see in our body. Some names that I want you to make sure you know is things that are epimerases are going to change the stereochemistry at a carbon. Things that are isomerases interconvert aldose and ketose sugars. Again, our case study on fructose metabolism, here's the important pieces that I want you to know, right? That fructose metabolism differs in liver versus muscle, right? And a key piece, and we're gonna elaborate on this later in the semester, but if you are using fructose as a fuel in your muscle, that's going to be fine. It metabolizes normally. But if you have excess fructose and it needs to be metabolized in your liver, why this is problematic is there's no regulation of this enzyme. And so you are going to rapidly deplete your ATP levels. So even though you are consuming excess fuel, by depletion of ATP levels in your liver, your body thinks it's in crisis. And so it's going to be like putting out all of these signals that are going to say, hey, let's eat some more. Let's suppress our leptin and activate our ghrelin hormones. Now, you don't need to know those hormonal pieces for this learning unit, but I just wanted to get them into your brain because it is something we're going to revisit later on in the semester. But I do want you to know the important piece is that fructose regulation or fructose metabolism in the liver is unregulated. But at the end of the day, it's a fuel that's in excess. So we're going to be making acetyl-CoA that we make into fatty acids, putting them on a glycerol backbone, storing in adipose tissue. Okay, that's the theme that we're going to see no matter what the excess fuel is. All fuels funnel to acetyl-CoA, so all fuels can become fat. Again, I keep hearing giant ice chunks fall. I'm defrosting that freezer, like hearing giant ice chunks sort of fall down. And I'm not going to go through too much of this because there's a great video that's on it that integrates these two slides. I would encourage you to understand this is a really important aspect of things from this learning unit. Thinking about how curves shift. What does it mean when a ligand binding curve shifts? What does it mean when an enzyme activity curve shifts? And be able to think about the concepts of what happens if I pinch a pathway, right? For this case, we knew specifically what these enzymes were. We knew that this was an upstream enzyme. We knew that this was a downstream enzyme. So be able to think about and understand this story so that you could apply this knowledge in a fictitious metabolic pathway you haven't seen before, okay? And I think, yeah, that represents sort of the end of our content from our notes. Is there anything that anybody wants me to go over in a little bit more detail? I can pull up the homework key if there's anything you want me to look at with homework. And again, this is, I don't have time to rewrite my keys. You guys are smart enough. You can sort of figure out if the question numbers are different, if I don't have something. But do use this as an opportunity to make sure that you understand sort of these different pieces. I'm just kind of looking through here. Obviously, we didn't talk about this. That's a structure that I don't talk about anymore. So you don't need to know that guy. You'll recognize a lot of this stuff. Again, we used to go way in more detail on the pentose phosphate pathway, but we don't anymore. Again, here's just that cascade sort of shown in a different way, 2,3-BPG story. And that's pretty much what your homework was. All right, questions? No, all right. Well, I'm around tomorrow. I know, let me remind myself. Those of you who are in 410, I think, yeah, Dr. Brown is just going to be talking about writing your introduction section. So those of you, right, who are my research students, you pretty much sort of have this done, because I think for the most part, like a good bit of your proposal might be done. So I might use this as an opportunity to continue working on aspects of your proposal that you that you need to have for biochem lab. So maybe writing your material or your methods development section, but that's all that we're doing. It's just a writing section session in 410. I will say on Thursday, those of you who are in 410, I'm going to talk about personal statements. It's really only going to be for about 20-25 minutes. I have a meeting that people asked me if I was available to start it earlier. We weren't going to originally start till 4, but I said, yeah, I probably can get through what I need to with you guys. And then Ms. Pat Note is going to talk about safety for a little bit. And that's probably going to be a class that'll be on the shorter side. Okay. And then we have another sort of writing session next week, Tuesday, another working session the following Thursday, and your first draft is going to be due by next week, Thursday. I think many of you who are in biochem are currently in 410 as well, so that's sort of relevant. But I wanted to remind myself, I didn't think there was anything that was really going on over the next little bit here with that. Anything else? All right. Well, I will be around tomorrow. Probably not until 11:30. I have to go get my haircut, important things like that. And then I'll be around. I know I've got some Gen Chem students that are going to stop by, and so forth. But and then we've got class at 2, but pretty low key. Okay, so stop by if you have any questions. All right. Good night, guys. We'll see ya.
Thank you.
You're welcome.
I have a question.
Yeah.
So I know I talked to you about meeting with you to go over the stuff that I missed on Friday, tomorrow.
Yep. Yep.
However, after going through everything, I am a lot more worried for the exam.
Okay.
So would it be better to like focus on exam stuff tomorrow and then like
Oh, yeah. Yes.
Wednesday during
Yep.
office hours maybe come in
Yep.
instead?
That's totally
Okay.
fine. Yep.
Okay.
Yeah.
'Cause,
No.
like I want to
Nope.
get material from Friday, but I also like really want to focus on preparing for the exam because after taking the quiz today, I realized like I was not studying in a way that was beneficial because I thought I studied well over the weekend for that.
Reminded me, I meant to say something to people that you should be able to see, because they won't be released until the last person takes the quiz, but everybody should be able to go on and see, like, not just their score. They should be able to see the question, and they should be able to see like what the right answer was and what they put. Are you able to see that?
I haven't checked it, but I
Okay.
was able to see like which ones are like I got wrong right off the bat. But let me just
Mhm.
see if I can Blackboard just wants to reload 50 times. Okay. Biochem 2 unit 2. Okay, wait, I need to go in the grade book. That's what it is.
Oh, and you know what? I'm gonna stop recording here.